Selective laser sintering, or SLS, is an additive manufacturing technology that uses laser to melt solid powdered particles and then to fuse them together in a desired shape. It creates objects with superior mechanical properties, complex geometries, so it is actually ideal for the creation of custom-made biomedical devices. However, it is still underutilized in this field due to the lack of suitable materials. So in order to overcome this issue, the aim of my PhD was to establish a platform that can create sinterable core shell particles, which will have sinterable polymer as a shell and non-sinterable one as a core, and then that core can potentially incorporate drugs or any thermosensitive material. Finally, at a later stage of my PhD, I wanted to use these particles to create biomedical products. As a first step, I established a platform for the production of particles which contain a device called Jet Mixer. You can see it on top of my poster, and in Q&A part I can explain you how it works. But what is important is that it is able to produce core shell particles with liquid cores, or nanocapsules as they are also called, through the process of flash nanoprecipitation, and it can also be used for the preparation of emulsion whose dispersed phase droplets can then be photocured, finally lead to creation of core shell particles with solid photocured cores. I tested that device for both purposes, and its big advantage is the ability to continuously produce materials in quantities up to several kilograms a day, which is a big improvement compared to microfluidics, for example. So that was the first step, the establishment of that platform, and then in the next step I proceeded to test its potential in the creation of core shell particles with liquid cores, which you can see in the results section 1. So I used different types of polymers. I proved that the device can be used to reproducibly create nanocapsules with sizes between 80 to 200 microns, which can be controlled through the concentration of the polymer. Finally, I chose one of these polymers, or PCl, as a coating material for my core shell particles. So you can see it in section 2. My particles contained acrylate material in the center, which was surrounded by a layer of surfactant pluronic and then a layer of PCl. Pluronic and PCl melt at the same range, around 60, so during SLS they will melt, particles will bind through that layer and the shell will remain unaffected. This structure, it was confirmed by TOF imaging, which you can see in image 6. How I prepared it, again, we can discuss it later, but what is important for SLS is that these particles contain spherical shape, as you can see in the results section 3. They had sizes between 50 and 70 microns, which is ideal for SLS, and also PCl as a coating material has ideal thermal properties for SLS, and it has previously been used for this purpose. It is also biodegradable and biocompatible, so great for the production of biomedical products. So finally, I tried to sinter these particles alone and in combination with another biodegradable material, pluronic, just to see how they can be used in multi-material and single material products. And I managed to successfully create objects which you can see, square-shaped objects which you can see in the image in the results section 4. So as a future step, I will swap this core material for a biodegradable one. I will incorporate model drug ibuprofen, and then I will test the solution on these particles. But the big achievement was so far like the proof of concept, the ability to create as particles which have several different zones, so core and 2 shell materials, and also the establishment of this platform which can produce large quantities of such materials, which is a big step forward for creation of new materials for biomedical SLS.